I mean, this bit of kit here, this is one of the workhorses of physical oceanography, right? This has been used to map the depths of the oceans. As it goes down, it records. As it comes back, it collects water samples. And you're using it here for measuring water much closer to the surface. What do you do with the samples you get out of this?
So the samples that we get out of it, we take for phytoplankton, taxonomy, we take to characterize the pigments, we take to look at the dissolved organic components, we look at the suspended matter, and all of those discrete measurements are coupled with the optical data so that we can turn an optical signal into a concentration.
And these, what the bottles have done, so they're open both at the top and the bottom, and as the CTD went down through the water at some point, or as it came back up, the top and the bottom snap shut. So what we've got in here is a water sample from different depths. So this is what the ocean is doing right below our feet. Exactly.
On the computer screen on the bridge, we can see the profile of temperature and fluorescence, and then we can target wherever we've got a feature, and we can close automatically the bottles at any specific depth that looks of interest to us.
So the water is now in those bottles. How are you going to get it out?
So firstly, I'm going to release the pressure using this tap here, and then I'm going to release the water by opening the tap here, and I'll then rinse the bottle so that I haven't got any cross-contamination from washing the bottles in the lab with milieu water. So I'll do that a few times.
So the crucial thing here is to make sure that what's in the bottle is as representative of that part of the water column as it possibly can be. It's not contaminated by anything else.
Exactly. So yeah, every week we use these bottles, but obviously they're washed using a washing protocol, and we don't want the seawater to get diluted, so I just rinse them out a few times in order to do that.
So as we look at the water coming out of the tap just here, it looks like water. It's transparent. It doesn't look like it's got any colour, but it does have a colour.
It does. I mean, as we look at it coming out of the tap, yes, obviously it looks transparent, but within every single drop of water, you could have 1 million phytoplankton cells. But not just that, the phytoplankton cells may shed their content, and that forms a colour which is dissolved that we can't normally see with the naked eye.
And this is the sort of thing that you're interested in, Hayley, the colour that all of these tiny components of this make out in the ocean.
Yeah, all of these individual different components that come from the phytoplankton or the detritus, when you look at them from a big scale perspective, like from a satellite, they all influence the ocean colour. But it's only through doing this that we can really understand exactly what's in any one bit of water in particular.
The process of satellite validation then, there's a lot of detail in here, in these water samples, that lets you cross-check with what satellite data actually means. How does that process work?
At the time of orbit of the satellite, the instrument will go into the water, we bring it back on board, we sample it into discrete components. So we've got the phytoplankton, we've got dissolved organic material, we've got total suspended material, and we want to be able to measure the optical signal from each of those components, but also the associated concentrations. So if we've got a clear day, which is not like today because it's just started raining, if we've got a clear day where we've got in the location of the ship, in that pixel, a clear image with no surrounding contamination from clouds, we know that at that particular point in time we're going to get a match up with the ship sampling to the satellite data, and it's at that moment that we can use the data from the bottles to then use for algorithm development or algorithm validation.
And Hayley, there's a lot of challenges associated with this, right? Apart from the fact it's just started raining, what are the difficulties in doing this? What are the things you've got to correct for and, allow in and make, cross-check? How do you make all this work?
Well, one of the problems with validation, and you can see that today, is that there's a lot of cloud. So if there's a lot of cloud, then the satellite can't see us and we can't see it from here. So we can't actually get a picture when it's cloudy like this. And so any data that we collect, we can't match up directly with the satellite. Now, in normal situations, if it's completely clear and the satellite can see the ocean surface, there's still the atmosphere in the way. So we've got to correct for the atmosphere, and we can take measurements from here as well, looking up, to help us do that.
So it's not just about what's in the water then, it's also about what happens in between the water and the satellite.
Absolutely. I mean, apart from the ship here, which is obviously our coastal-based platform, but at Penley Point, just around the coast, we have an atmospheric station, and we're taking measurements of the atmosphere and the type of particles in the atmosphere that are used for this model of atmospheric correction, which is really the hardest thing to do when we're actually talking about validation.
So it's all these nitty-gritty little details, but then once you've done that, you can be confident that the data that comes out of the satellite process and the data that's available to the public for the Sentinel system, for example, does actually mean what it's supposed to mean.
And that's when we can start using it for applications. So all the different components that Gavin has mentioned, I think like the dissolved organic matter, the different types of phytoplankton, are very intimately linked to the quality of the water here. So if you're fishing here or you're swimming here, you want to know that that's good, safe, clean water to swim in or to eat food from.
And what sort of biodiversity do you get? You were talking earlier about lots of different species, so phytoplankton, these tiny little plants. There's lots of different types. And how much does that change with time? Is it always the same in one place?
Well, here at this coastal site, we get two blooms. One in the spring that is normally often dominated by diatoms, but it also is co-dominated by Phaeocystis. Phaeocystis is a problem species. It is linked to eutrophication, so that's high nitrogen that runs off from the land. It occurs as tiny small swarmers, but then as it develops, it forms gelatinous colonies, and these gelatinous colonies form foam that wash up on the beaches. Then towards the late summer, we get a secondary bloom, and it's in this secondary bloom that we can often get dinoflagellates, which cause harmful algal blooms that can either shed toxins into the water column, or they can strip oxygen out of the water column, which is problematic for fish and aquaculture.
So there are these plants, lots of species are living in the water column all the time, but because perhaps there's an injection of nutrients or the temperature changes or the weather conditions change, you sometimes get a bloom which is a big aggregation all growing at the same time, and that can either be beneficial for the local ecosystem or it can be harmful.
Absolutely. Each phytoplankton species is adapted to a range of temperature. So as we continue through from the winter to the autumn, you get a natural step change in the range of which phytoplankton species are adapted to their optimal temperature. And of course, overlaid with that, we might have weather conditions where we get a lot of rain, which is washing nutrients out of the rivers and inputting them to the coastal zone. And then you have this competition between the phytoplankton species that are adapted to this range of temperature in terms of utility of the nutrients that they are optimal for them to bloom.
So you can make these measurements and you can compare with the satellites out in the open ocean, but Hayley, you're especially concerned with the coasts. And the coast it's a challenging place to work for satellite data. Tell me about some of the it's interesting because that's where the people are, right? People care about things at the coast, but it's also a very difficult place to make satellite measurements.
Yeah, it can be quite difficult, and one reason is that the coast is has a very sharp coastline, so you've got to be able to get close in, look at little bays, look at beaches, and that can be quite difficult because we don't always have the very high resolution data that we need. With some of the new satellites in the Sentinel series, we're beginning to get this, which is really great news for us when we want to use these applications. The other things are we've got to be able to differentiate between what is land and what is water, and for us as people that's easy, but from a colour perspective that can be quite hard, especially if you're in an estuary or you're in an area where there's a lot of tides and you don't know if it's water or if it's dark mud or things like that. So it's a very challenging environment to work in, but as you say, a very rewarding one because that's where we interact with the sea itself.
And so the sorts of places people would use this data are not just fishermen, but people who are maybe planning where ships are going to transport goods. What sort of applications does it have?
Lots of applications, as you say, fishing, aquaculture, things like that, but also things like planning, deciding where you're going to put a marine reserve, or where you want to put a new wind turbine, or where's safest to have your ships come into the port.
There's lots of different applications. It all starts with little bits of water coming out of bottles like this. Measurements like this are very valuable in one individual location, but there's something special happening here, which is that the same measurements have been made for a very long period of time. Tell me about that.
Plymouth Marine Laboratory have been making measurements here. In fact, the measurements go back for 100 years at the same site.
It's astonishing. It's astonishing.
Astonishing, yeah. The physical parameters, temperature and salinity, go back 100 years, and obviously are a unique record of change for this coastline. Going back to 1988, that's when we started to characterise the biology in any greater detail, and since then we've got information on the carbon compartmentalisation into different phytoplankton fractions, sizes, species, right up to the zooplankton level. And with colleagues at the Marine Biological Association, they also look at the small fish. So it's over 20, 30 years of continuous biological data at this site as well.
And that's really important because you can see how a single site is changing, and that's so rare, isn't it? In the oceanographic record, there are very few places where we say at this spot 50 years ago we know what it was doing and we know what it's doing today. So what are the benefits of this kind of time series, Hayley? It's very rare. It's obviously people have put a lot of effort into it, but why bother?
Well, one of the good things about having a long-term time series is that you can start to figure out not just what is happening, but also why it's happening. And then you can attribute those changes to different driving forces. So you can start answering questions like, is this related to climate change? Or is this because, we built a new town there and that's causing more nutrients to enter the water? And those sorts of things are really important for when we decide how to manage our interactions with the marine environment. And this is the same thing that's now happening with the satellites. We've got these long time series of in situ data, and with programs like the Sentinel series, we're looking at getting long time series of satellite data as well.
And it matters a lot because the natural environment varies quite a lot on a week to week or month to month and even a year to year basis. There's quite a lot of noise, it goes up and down a little bit, and that's just natural variability. But what we really want is to extract out the big patterns, and that's what you need the time series for.